Good morning, everyone. I know we still have a few people logging on, but we're going to go ahead and get started. So good morning and welcome to the 2022 CRB Webinar Series. Today's webinar will provide an overview of upgrades and improvements for the production and distribution of clean plant units at the Citrus Clonal Protection Program. I am Caitlin Stanton with the Citrus Research Board. Today, I'm joined by McCall Machado and Dr. Joey Mayorkin, who will be running the polls and monitoring our chat, as well as Dr. Melinda Klein, who will be introducing our speakers. Please note that this webinar is geared toward growers and pest management professionals. Master gardeners can certainly benefit from participating, but the pest management methods presented, especially the pesticides, should not be followed without a clear understanding of their legal use in home environments. And we encourage you to ask questions during the webinar using the Q&A function, which can be found at the bottom of your screen. The speakers will answer all of the questions following their presentations. And with that, I'm going to pass the mic over to Melinda, who will introduce our speakers.
Thank you, Caitlin. I'm very happy to be welcoming a UC Cooperative Extension Farm Advisor, Dr. Ben Faber. Dr. Faber has served as a farm advisor in Santa Barbara and Ventura County since 1989, covering issues associated with pests and diseases of various crops, as well as the issues of managing soil and water to improve production and reduce soil and water degradation. Dr. Faber's emphasis is on subtropical tree crops such as citrus, avocado, and cherimoya. Today, Dr. Faber will provide a regional update. Ben, please go ahead and begin your presentation.
Yeah, thank you very much. You know, one of the major issues in life is how to tell what's going on with weather, and you know, we just don't have a very good handle on what's going on with weather. The major problem in irrigated agriculture in California, by my estimation, 80% of the problems are related to irrigation management. And you know, if we knew what the weather forecast was, we could better manage our water. So the forecast last fall for this area was that it was going to be no rain, hot, miserable, and you know, here in Ventura County right now, it's kind of cool. It's 65 degrees at 10 o'clock in the morning. And we had a heat dome that was supposed to blow through here and nailed San Francisco and completely missed Ventura. You know, we had a high of like 85 degrees in Fillmore. But, you know, the heat is coming, and what is an issue with heat is that it affects how we manage water, which means how drought affects plants, and salt stress. And so the water that we put onto our trees is loaded with salts, and we got to manage those salts. And you know, fortunately, a lot of the salts in our waters, our groundwaters, are laden with calcium, which is good, but there is some sodium and chloride and boron, which is a real problem. And so we got to manage those, and it's got to be in an imbalance with the nutrients that are in the soil. If we put on too much water, we're going to leach nutrients. So we can manage salt, we can manage heat, we can manage drought effects on plants by managing our irrigation water. And I'm bringing this up because it's not that it's a hot topic today, it's a hot topic every day. You know, so we're going to go through some of the issues. So the last 20 years have been associated with these heat spells, and the darker the coloring here, means the hotter it's been, and for a longer period of time. And unfortunately, if you go back into the record, there's been periods when we've had 200 years of drought, when we're getting not less than 20 inches, but we're often getting less than 4 inches per year. And this is the forecast, or the current state of drought in the West. This is from just 2 weeks ago. And again, you know, forecast is that Central Valley is in deep doo-doo. That extends down into Ventura, Santa Barbara counties. So we've got to learn how to deal with heat. And so I bring the heat up because it affects trees in different ways. And we were getting calls, I was getting calls, 2 and 3 months ago, problems. And, these, we've all seen these issues of, wilting of trees. This is boron toxicity. Citrus is very susceptible to boron toxicity. And to a certain extent, you can moderate that with the rootstock that has been chosen. But, all citrus is prone to not just boron, but also salinity and dieback. Dieback is caused by a stress, salt stress, heat stress, water stress that induces some death that occurs, and Botryopsphera fungi jump on that, and they can actually grow right down a stem and on a 1 to 2 year old tree, kill it. But you'll see these trees with, a few dead leaves or branches on a tree, and it goes back to a water stress issue. So we'll see fruit drop, and we've already seen some fruit drop, fruit cracking, which is often just associated with wet, dry. You get behind your irrigation schedule and you irrigate and boom, the fruit blows up. And these Botryopspherias that I was talking about, the diebacks, can actually go to fruit and cause fruit decays, as well as dried out fruit. So fruit that sits on a tree a little too long can end up with this, what's called endoxerosis, which is dried out fruit. So what do we do about stress? Water stress? Well, we fix the irrigation system. The systems that we have out there, tinker toys, they break down, they fall down. When you're tripping through the orchard picking fruit, the coyotes are chewing on it, gophers are chewing on it. We need to learn how to apply water uniformly. And this again goes back to an irrigation system evaluation and scheduling irrigations, and then applying a leaching fraction. And this has always been a black hole. How much water do you apply to remove salt that is, leads to Botryopsphera, that leads to wilting, that leads to boron and salt toxicities? How much do you apply, and how do you apply it? Well, this is a really big question, and it's going to lead up to a seminar on its own, at least a blog and a newsletter article. But how much water do you apply to leach? And so you can do this through actually measuring soil salinity, and the soil salinity is always going to be higher than the water. So our waters typically are running 2 to 3 decisiemens per meter, which is 1,200 to 2,000 part per million salinity. Well, the soil is always going to be higher. It's going to be running 3,000 to 6,000 part per million. So when the tree pops water out of the soil, it's going to leave salts behind that concentrate, and that leads to salt issues. So we need to know what a leaching requirement is. And in the past, we've sort of said, well, you got bad water, put on 15% more water than the tree needs, but devising a leaching requirement in the past has been kind of like shooting in the dark. And there's been this issue of, okay, we are adding water, and as we add water, okay, the top layers are where most of the roots are. And as water moves down through the soil, it creates leaching. But what happens is that as it goes with depth, the salinity increases with depth. So it gets to a point where in some soils deeper down, may be so saline that the trees can't use that water. So the other issue is that most of our leaching recommendations have been based on flood and sprinkler. And so we really haven't devised recommendations based on drip and micro sprinkler. And so what we need to recognize is that when we're irrigating with furrow and flood and irrigation, we're actually taking water and it's being pushed straight down more or less. Whereas in drip and micro sprinkler, we're getting this wetted pattern where we get water here, but you tend to move salinity out to the margins of the wetted pattern. So if you look at the pattern as water moves out from the emission point, we get this wetting front where salinity is carried. So how do you adjust your leaching requirement based on this pattern that develops in the wetted zone. And so here's a really clear example. You know, we've got, this is in grapes, and so the water is being dripped down, and the water, you know, dissolves the salts here, and as the water moves out in the wetted pattern, you get this zone of salt accumulation. And plants can't take that up. It gets so concentrated that, you know, it may be wet out there, but it's so saline that the plant can't take it up. And so the amount of water that you apply is going to be based on the soil texture that you have as well. So as the coarseness of the soil becomes finer into a clay, it holds more water, and we can put on more water that supplies what's called the field capacity. But that's going to adjust how much water we're putting on to actually cause leaching fraction. So this is all leading up to a discovery I made. Laoxiang Wu at UC Riverside put together this pretty easily managed website. It's found at this address, salinity.ucr.edu. And you put in the crop. Okay, so in the case of the crop, we put in citrus or avocado or something like that. You put in the irrigation water quality. So you put in 2 decisiemens per meter, 2.5 or 3, or gosh, I hope you don't have 4, but you know, waters in Ventura County are terrible. You know, they're groundwaters. We've got saltwater intrusion in certain areas. We've got high calcium sulfate, which is a salt, even though calcium and sulfur are beneficial nutrients. So you put in the water quality, you put in the soil texture. Okay, this is going to determine the amount of water holding capacity. You put in the irrigation system, whether it's drip or micro sprinkler. It lists flood and furrow irrigation too there, but you put in what irrigation system you have, and boom, it's going to kick out what your leaching requirement's going to be. So this is the model, and here is an example. We've put in lemon. We put in 3 decisiemens per meter, and we can actually put in the rainfall. So if we've had 5 inches, 10 inches, in some areas in Ventura, we actually had 12 inches this year, which was forecast to be a low rainfall year, but we had that rain that came in December, and whoosh, it was so great. We really are dependent on rain to leach the salts from the accumulated salts that occur from our irrigation applications. We put in the soil texture, loam, we put in drip irrigation, and it kicks out. We need a 21% leaching fraction. So, so it actually will tell you based on ET, the amount of water, and it will actually tell you the amount of water per acre to apply. So this is a really neat website. I encourage people to use it, if only to get a better appreciation of how much water to apply to accommodate for removing salts from the root zone. So you can learn more about this from our newsletter. Here's the website. And we have a blog that comes out 2-3 times a week talking about issues like irrigation, like botryophoria, like salinity management, and a lot of the other issues that citrus growers face. So that's what I have to offer today, and good irrigating.
Okay, well, it does look like we had a quick question in the chat. How critical is the irrigation efficiency?
Ha! Irrigation efficiency is measured. The less water you put on, the more efficient it becomes. But really, irrigation, so it's water required over water applied. So the less and less water you put on, the more efficient you are. But that's not what you want. You want to put on what the crop needs. And I always preach this, you know, you're looking for a healthy tree, not so much as saving water. You want to put on the amount of water required for a healthy tree. So irrigation efficiency is really not a good measure. The measure of the application distribution uniformity. So we're talking about, you know, the amount of water that's evenly applied over an orchard, you know, the different emitters, how efficient, how close to the average those emitters are putting out. And then how much water is required based on evapotranspiration. So the closer we can meet the needs of evapotranspiration and the leaching requirement, the more efficient we're going to be. So irrigation efficiency is not really a good metric, as they say, for measuring applied water.
Okay, thank you. Hopefully that answers the question from the audience. And so with that, thank you, Ben, for sharing that information with us today. I know we've got more heat coming on, so irrigation is always important this time of year.
It's coming.
Yeah. So now I'd like to welcome our today's speaker, Dr. Georgios Vidalakis, Director of the Citrus Clonal Protection Program and Plant Pathology Extension Specialist for the University of California, Riverside. Dr. Vidalakis' research program focuses on the etiology of putative viral diseases, as well as the ecology and molecular biology of citrus-infecting viruses in California. As director of the CCPP, Dr. Vidalakis is responsible for overseeing this internationally recognized program, which provides growers and researchers with stocks of stable, disease-tested citrus fruit and rootstock varieties. Today, Dr. Vidalakis will discuss the preventative and multi-layered citrus disease management approaches with a focus on activities of the CCPP. Georgios, please go ahead and share your screen to begin the presentation.
Thank you, Melinda. Screen number 1. Okay, I have the controls. Welcome, everyone. My name is Georgios Vidalakis. I'm responsible for the Citrus Clonal Protection Program. And as you will see throughout my presentation and from this introductory slide, the CCPP is a collaborative program between the university, the regulators, and most importantly with the industry. And yeah, okay, so it should be unfrozen. So first, I always like to start my presentations telling the audience that what you're going to hear today by no means is 1 month's work. There's a large group of people working with me at the CCPP, and we have collaborators throughout the state, the country, and actually the world that we are all trying to create healthy plant units for citrus propagation. So today I'm going to give you a brief background for the CCPP. And as we are going through to set up the stage, and as we're going through the core functions of the program, we're going to talk about the 6 important upgrades and improvements we have made. And while we're going through the presentation, keep an eye on the slides. The Q&A inside the box will appear on the top right corner of the slide. So when you see that in a slide, focus, maybe grab your pen, keep notes. This slide will be related to the questions for the credit. Okay. So the keyboard works, the mouse doesn't work. So it's illegal, it's prohibited. Citrus is prohibited from entering the United States as Budwood or as a tree. This law was passed in the 1950s to prevent the introduction of catastrophic diseases into the United States. But how are we going to grow citrus in the US? The citrus is not native to the Americas. We have to have a mechanism to bring it in. So the regulations continue, and they say citrus can be introduced in the United States under a specific permit. And today there is 3 programs in the United States that have such a permit: the CCPP, the USDA Germplasm Repository for Citrus and Dates, and the Florida Budwood program, the State Department of Agriculture in Florida. So we are these 3 programs that primarily bringing in citrus into the United States and make sure it becomes available to the industry and the researchers. So our programs operate at the forefront, at the exclusion layer for plant diseases. It's the best plant pathology measure against any disease. You don't have a disease, you don't have to worry about it. And you can see how as we're moving down in the principles of plant pathology, you get a disease, you try to eradicate it first. how we're moving from an investment of $250,000 a year into tens of millions of dollars. We spend roughly $45 million dollars a year in California to chase the Asian citrus psyllid. And then when you have to live with the disease and you have to transition to management, that's when really the losses for the industry become in the billions of dollars, like what's happening with HLB and Florida. So our programs are preventative. And the way that you get a disease is when three factors come together at the disease triangle. The environment is conducive, the pathogen is present, and susceptible host is present. So at the CCPP, we cannot control the environmental conditions, but we can control the pathogen. We make sure we remove pathogens from the propagative materials, and we make sure we make available hosts to that they do not have the pathogens, plant material to be planted healthy in the fields. So we make sure that the pathogen is not present in the host to cause disease. And this process of diseases threatening the industry and us helping has been going on for decades. So in the very early years of the industry, cirrhosis, a virus disease, was a big problem. And the solution was grow nucellar seedlings, grow citrus from seed. But the seedlings had issues with Phytophthora, so the industry switched to using sour orange rootstocks. The problem with sour orange rootstock was that it was sensitive to CTV decline, tristasia decline, so the industry shifted again around the 50s, 60s to changing the rootstocks to trifoliate and trifoliate hybrids. And trifoliate hybrids are sensitive to viroids like the exocortis viroid. And who knows what we're going to get in the future that would be resistant in terms of scions and rootstocks to HLB or other exotic diseases. So in every turn, and these are massive turns, imagine the whole industry switching rootstocks and scions. If in any of these terms you don't have a source of clean budwood, then you end up switching one problem for the other. And it has happened in other countries and regions in the world where they didn't have programs like the CCPP. Whenever they made those switches, they ended up solving one problem and then they traded for another. So our story goes back in the 1930s when Dr. Fawcett discovered the first sick tree with cirrhosis and made the observation that if you harvest budwood from a cirrhosis tree and you make trees, new trees, propagate trees, the other trees will also have cirrhosis. So the industry with the university and CDFA started the cirrhosis freedom program in the 30s. And you can see how more or less every 20 years, the last few years, we have accelerated the progress of the program. There is a transition to the program. So we're constantly evolving. And one very important transition that happened in 2009 is that the federal government realized the value of programs like the CCPP and brought all of us under the umbrella of the National Clean Plant Network. And the program is focusing exactly on detecting pathogens, removing them from the germplasm, and distributing this material to all stakeholders. And this is happening for many different crops, not only for citrus, all the vegetatively propagated specialty crops. And the network is very large and includes many commodities, and it covers the whole nation. So why do we make this huge investment, and why all these commodities want to do this? Because we want to avoid people from traveling around the world seeing an interesting citrus variety or an interesting cherry variety and put that budwood in their suitcase and bring it back home. That's not the way to do it. Not only it's illegal and it's prohibited, but the risk for varieties of the risk for introduction of diseases is great, and is causing downstream issues with putting pressure on established industries. All of you have experience when you travel to island nations or islands in Hawaii, or we go to Australia. They have very strict rules about people moving vegetables and plants in and out, because that's how pathogens move. The humans are very efficient vectors of plant diseases. So to look back to the CCPP, as we said, we're a very collaborative program between universities, stakeholders, the industry, and the regulators. And we do two things. We make sure we have a safe mechanism of introducing varieties into California and the United States. And then after the varieties have been processed, we maintain foundation facilities and blocks, and we distribute the material. We are achieving our two main goals by doing those 6 things. We bring bacteria in, we detect pathogens, we remove them, we establish foundation facilities, we retest the foundation trees, we distribute material, and what we're doing today, we make sure we communicate with the industry to know about the mechanism and how they can access our material. This is a very old slide I made years and years ago, but more or less give you the function of the CCPP. On the right-hand side, behind the red line, is what's going on in Riverside in our quarantine facility. And after our therapy and diagnostics procedures are completed, the material crosses the red line, crosses the quarantine, imaginary quarantine line, and enters the foundation facility at the Lindcove Research and Extension Center in the heart of agricultural production in California, and from there finds its way to nurseries and growers. And then if we were to see the CCPP functions in a flow chart, the functions appear more or less like this, where between every step, there is a test, and if the test is negative, the material is moving forward. If the test is positive, it's looping back to therapy until everything has tested negative and we're releasing from quarantine and into our foundation facility. So one of the first improvements we did was to build skill and capacity for our personnel. In 2005, when I started as a director, we were 4 or 5 people, and there was no way that the program could move forward and meet the challenges and the growth of the industry on this size. So we had long conversations with the industry and the university and the federal and state agencies, and now we have a well-staffed program with all the expertise needed to meet the growth of the industry and the challenges of the future. We have a healthy budget, a healthy number of people, and we are operating at 3 facilities, at the Ruby quarantine facility, at the Lingo Foundation block, and we have our molecular biology laboratory in the main UCR campus. So after we took care of the personnel issues, we focused, or actually in parallel, we focused to bring our facilities where they needed to be, because not only we didn't have the capacity at Riverside and Lindcove to do our job, but we didn't meet any regulatory standards, especially with the HLB threat approaching the state of California. So for the facilities in Riverside, this is what I found when I started as a director in 2005, facilities that they were primarily built in the 1950s. So we start eradicating all the transite that was there and all the leaky roofs, and we set up the foundation, and we built a nice functional head house to help with the therapeutics and the diagnostics at the Ruby quarantine glass house. And then we inherited this screen house that was built in the 50s, wooden structure. It was a beautiful structure, but it could not be upgraded to keep the silids or the aphids out, and we had to demolish it, and we put up a beautiful stainless steel frame, modern greenhouse, so we have bench space capacity and also keep pests out. Also, our equipment was completely outdated. We were trying to do tissue culture and therapy in wooden boxes with fluorescent lights and said cloths. So again, with the support of the industry and other funding agencies from the Citrus Research Board, the Citrus Nursery Board, and the NCPN, we're able to upgrade completely our equipment and being able to perform our tissue culture and therapy duties. So specifically for the 2 core functions of therapy and diagnostics, I'm going to show you a couple of cool items here. So when Budwood arrives at the CCPP, it's placed in culture in media, and then after a couple of weeks, the budwood is flushing, is producing new growth. And then if you were to sit in the microscope and look under the microscope, you will see exactly. So the blade that it's moving here is this size over here. It's less than a millimeter. And our technician here is removing, I see the video stalling a little bit, is removing leaf primordia to expose the apical meristem of the plant. And let me see if I can advance the video a little bit. And this last little leaflet that was removed is microscopic, you cannot see with naked eye. And essentially we are removing the top layer, a few cells of the apical meristem, and we're going to propagate it on a little rootstock and regenerate a whole plant from those cells. The point here is that from this little mass of cells over here, we start growing out a new plant. And the key here is that those cells, because they are what we call undifferentiated, they cannot support the replication of pathogens. So we are avoiding, by making this cut, we're leaving the pathogens behind in the rest of the plant, and we're using these healthy cells to recreate a plant. After a few weeks in the tube, the plant looks like this. We're picking it out and we're propagating it. We remove the roots and we're propagating on a mature rootstock. And we put it in a growth chamber, and a few months later, actually weeks later, maybe a couple of months, this was the original rootstock that you see bent here, and this was the rootstocks inside the tube, and then the variety is growing out. And after the trees have produced enough tissue, we move them to therapy so they can be removed, released from, we are moving it to testing so they can be released from quarantine. So with these improvements, we have built capacity to our program. We can bring in a lot of varieties, we can therapy a lot of varieties, and we can release from quarantine a large number of varieties. And part of this effort also has accommodated a big collaborative effort to bring material from other states like Florida that they may have, because they're dealing with the HLB epidemic and they have seen how different varieties and rootstocks behave under the natural spread of the disease, maybe there is resistance and tolerance in those areas. So we have been bringing in from Florida, because now we have the increased capacity, germplasm that may be useful to us down the line. So we address the capacity issue, but then when we start looking how well we're doing, how fast we're moving varieties out of the program, you can see how the bars are getting compressed towards the 25, 30 months zone, but still we have a lot of work to do. We want to be able to produce germplasm in less than 2 years if possible, and that's the 104 weeks bar here. So when we start looking at these graphs, we realize that we lose a lot of time in this gray zone, which is waiting for the plant to grow. And as you will see later in this yellow zone here, which is where testing the plant. So the next effort was, for in terms of improvement, was to grow plants and trees faster and sustainably, and then move to improvements of diagnostics. So on this topic, we came in contact with scientists that they are thinking about those questions, and they introduced us to the concept of circular economy. So right now, the agricultural economy is linear. We produce food, and then we have waste that ends up in landfills or elsewhere. So the idea of the circular economy is that you take the waste, you are adding value to it, like a company that takes the almond cells and burns them in high temperatures in the absence of oxygen and produce biochar and pyrrolinic acid, or PA. And then by introducing these compounds back to soil, for example, you have an increased capacity of the soil and maintain moisture, and surface is increasing, and microbial organisms are getting increased. So we have a research project where we are trying to using these compounds for agricultural waste, and preliminary data are very promising. The addition of biochar, for example, in our pots where we grow our trees, so increasing bacterial populations. And it shows in this graph on the right, increase in seed germination, for example. And in a more complex experiment where we use the PA and the biochar in different combinations, if you just focus on the first two bars, the black and the red, you can see that presence of biochar increases the height of Carrizo rootstock, for example. Why PA, at least the way we used it in this experiment, had a small phytotoxicity down here on the table. You can see a small reduction of the tree height. So these are ideas that we want to bring in our program and make trees, grow faster in the pots in the greenhouse. Another concept that came our way was use shipping containers to create a perfect growth environment for plants where you control absolutely the light and the nutrients and the temperature and so on. And when we do a preliminary experiment simulating conditions like that, you can see in B how plants grew citrus plants in 6 months in the greenhouse versus in A in simulated conditions in growth chambers and incubators. So after a long effort and waiting for a couple of years, we finally received our MPGU, our Modular Plant Growth Unit at Rubidoux in March of 2022, and we're looking forward to start using it, growing plants, and have all of you visit with us to see what this new system can do. So as I told you earlier, we focused the slides we went through on how to grow plants faster and more efficient, but clearly we have delays because of our variety index or diagnostics pipeline. And that's where we focused our efforts next, to become more efficient in diagnostics while we're maintaining a high risk management level to exclude pathogens from entering the nursery pipeline and subsequently the orchards. So what's going on with diagnostics at the CCPP in the last few years has always been happening as new technologies arose from the 1930s and visual observations for cirrhosis symptoms to the biological indexing in the 50s. Then the immunological, the antibody technologies and molecular biology came into the picture in the 70s, 80s. And then in the 2000s, we have brought in the new quantitative or real-time molecular biology technology like the polymerase chain reaction. And where we're going to focus today is the high-throughput sequencing, which is going to be the future in all diagnostics, humans, animals, and plants. So the biological indexing, which is the strong foundation for quarantine programs, we are doing research on how different spectrums of light may affect symptom expression. So my PhD student is setting up those experiments with different ratios of blue and red light. And so far, she's getting beautiful results. Not only we don't get phytotoxicities and the plants growing nicely here on the healthy control, but we're also getting very delicate viral symptoms that we need to see in our controls to make sure that the test was valid. And when we start running statistics and replicated experiments, we actually start seeing, in this case, citrus virus A, which is a very ephemeral symptom, and you may miss it in the greenhouse. Not only more plants showed more severe symptoms here in this 95 red, 5% blue light, but also we saw symptoms almost 2 weeks earlier, 43 days versus 2 months in the greenhouse. So this is a very promising line of work that will help us to streamline the biological indexing processes in our program. Then we are constantly working on improving our multiplex capacity with PCR technology and universal detection of pathogens. And as I told you earlier, we are working on bringing this high-throughput technology sequencing in our program. So whenever a new technology comes up, there is an excitement and let's start using it. And this technology is so important that we want to make sure we do everything right. So we came in contact with the Oklahoma State University that they are pioneering their institute there for plant pathology forensics. It's world-renowned, works with FBI and other investigators on all sorts of forensic technologies. we came together and we said, okay, how can we solve the bottlenecks with this technology? How can we avoid having bioinformaticians all the time looking at the screen, trying to figure out if there is a pathogen there or not? And how can we make a friendly user system so anybody can use it in the Citrus world? And we came up with the idea that we are going to target a couple of different pathogen types, a virus, a viroid, and a bacterium that represents the big classes of pathogens in citrus, and see if this really can work for citrus. And a few years before this technology, to give you an example why this is very important, was a publication about a technology called MPGU. And the publication was very strong. All the work was impeccable. And according to the experiments, this PCR could detect up to 50 different pathogens in one reaction. So essentially, you can run one reaction at the CCPP, detect all the pathogens in one afternoon, and you're done. So when we put this technology to work, and we sent real citrus samples to the lab and so on, it came up that the validation of the method was done actually with synthetic targets. Nobody had bothered to run the technology with real live plant tissues. So the technology suffered because in a plant extract, there is sugars and proteins, and so it could not be applied. So again, we wanted to avoid issues like this. We wanted the high throughput sequencing technology to be applied properly, validated, and used the right way, because no question, as I told you, is the future in all diagnostics, not only for citrus. So we work very closely with the Oklahoma State University partners, and we set up our pipeline to evaluate how we're designing the probes that are going to look inside the database for pathogens, how we're going to do our initial testing, and how we're going to evaluate the sensitivity and the specificity and so on of the diagnostic assay in comparison to existing technologies. So after we figure out our pipeline, we target those 3 different kinds of pathogens that can be transmitted with Budwood. And one key point here is you can see the genome size. So we have very large viruses in the tens of thousands of nucleic nucleotides. We have viroids that they are less than 300 nucleotides, and then we have bacteria that they have millions of nucleotides in their DNA. So now, because your target is from very small to very large, you have different opportunities to make different sizes of electronic probes, and you can have a larger or a smaller number of targets that you're looking in your data sets. So these 3 pathogens gave an opportunity to put this technology to work. And a big problem with bioinformatic analysis of high throughput sequences data is, where do you draw the line? If I have one piece of DNA of a pathogen, do I call my sample positive so that tree has the pathogen, yes or no? And these are very tough questions to answer just by looking at the computer screen. So we came up first with simulated experiments. So we planted inside the data set different numbers of the target sequences, 10 sequences, 6, 5, and so on. And you can see that in this zone, the different probes, this is for the virus, this is for the exocortis viroid. There is an area where the computer starts getting confused. Is it positive? Is it negative? It's flip-flopping between positive and negative. But there is a point after a number of targets, in the example of Tristeza here, if the sample had 226 pieces of Tristeza inside in the millions of data in the data sets, anything above 226, the machine was very confident that the sample is positive. So we're able to run millions and millions of simulations and identify those cutoff values of what we can call positive and negative for a virus, viroid, and a bacterium. And then we put the system to work with real samples. These IPPN numbers here stand for introductory plant propagation number. These are actual varieties that came into the CCPP. So a CTV positive sample, according to a PCR result, also was positive for CTV with the probes we had designed for high throughput sequencing. In mixed infections, any single infections. Both the viroid and the virus probe work well. The same thing with the bacterium. And the healthy controls were always negative. So it looked like that the system worked very well. So we start using this technology officially in our program, and we held a workshop with the regulators and we explained to them what we have done. And for the last couple of years, we have been using high throughput technology for these 3 pathogens in our pipeline. And as I told you earlier, there is always going to be new technologies, and we cannot abandon one for the other. We cannot say, oh, the high throughput sequencing is the future. We don't need any other test. We're going to run high throughput sequencing and we're done. No, the risk at the exclusion level is very high, and we're always using complementary tests. So we have our biological indexing, and we're working on it to optimize it, as we talked earlier. We have our molecular biology on the second part of the table here, protocols, and we're trying to optimize them, combine them, multiplex them, always reevaluate our primers and probes. And yes, we do have also a new powerful tool, the high throughput sequencing, that we are developing, validating, evaluating, and we're bringing it into the family of tests. So when we're releasing a variety from quarantine, we're minimizing the risk for that variety to have a pathogen. And all these improvements that you've heard so far and upgrades have had this impact on our program. So the line, the orange, the thin orange line, you can see demonstrates the capacity. We have built the capacity in our program. You see how it's keep going up, number of varieties we can release from quarantine or introduce and release from quarantine per year. And at the same time, the little symbols, the little dots all over the graph, you see how they have been compressing towards the most recent years. And that's the months in quarantine, which is on the axis on the left-hand side. So we have increased our capacity with the improvements of personnel, facilities, technologies, tools, and we have compressed our time for releasing variety from quarantine with all these technologies, including modern diagnostics and so on. And there's still, you see, there's still some spots, they have been compressed, but there's still some spots over the 30 months limit there, the green zone. So this process is going to be always happening in the CCPP as it happened in its past, also into its future. So we address this first part of our pipeline, what happens in quarantine in terms of improvements and so on. So what happens after the variety leaves the quarantine facility and moves to the Lindcove Foundation facility? And that's the last 2 improvements we're going to talk about. So our foundation block also has been active since the 1960s. Unfortunately, in 2015, we had to transition from the outside block into the protected foundation facility. But this is a very important thing to remember. The citrus industry, and that's to tip my hat to all the members of the industry, how you have always been proactive. You didn't wait for the HLB crisis to happen and then move your nurseries and your Budwood sources indoors. You could sit down the line. So the screen house to protect our Budwood sources at the CCPP was built in 1998, almost 10 years before the first psyllid appeared in San Diego County. And then it was expanded the next year. And then in 2010, as we could see the threat approaching, we expanded. We built a second structure so we can have duplication and more square footage. And in 2011, we built a positive pressure greenhouse to duplicate one more time the Budwood sources under even more protected conditions than the screenhouse. And here's a few pictures of the 2010 construction of the screenhouse and the 2011 greenhouse. And now our greenhouse is full of replicas of all our Budwood sources. If a rip happens in the screenhouse, we have trees ready to go to replace after we make repairs and so on. And these events did take place in 2016. We had an opening in our roof, and we missed just a few weeks of Budwood operations, but we were able to keep supplying Budwood to the industry. A big improvement in our foundation block is that we moved to a a barcoding system, so we can track a sample and a Budwood sale from the mother plant all the way to the end user. And everything, there is no more paperwork, no lost notes or faxes or telephones, how it was the system a few years ago. Everything is recorded in the database. And because now everything is electronic and we have online ordering systems, the Budwood is really flooding and flowing to several thousands of users throughout California. Even though there is only 20, more or less, commercial propagation citrus nurseries in the state, there is actually thousands and thousands of people that are using clean Budwood for their backyard trees and so on. And this became very obvious. It was very clear when we found the first HLB tree in Los Angeles that had 23 different grafts on it and nobody knew where they had come from. So as I say in my talks, not everybody has a Cabernet Sauvignon in the backyard, but at least in South California, everybody has a lemon or a lime tree to cook and enjoy an afternoon drink. So you cannot take away the cultural connections of the California communities from citrus trees. So we want these communities to have access to clean Budwood so that when they make their Budwood sources, when they make their backyard trees, they are doing it with the right stuff. And as I said earlier, there is all these online systems we have created, included online ordering system for all the industry and all Californians. And we also have a system for all of you to make a request to introduce the variety from around the world. This is our variety introduction system and the Budwood online system. And then we are working on bringing all the system together under what we call LIMS system, a quality management, laboratory information management system. So this is another upgrade that has happened organically to bring all our efforts under one umbrella so we can analyze data as the graphs you saw and identify bottlenecks in our process and make sure we have the accurate records, to keep improving our services. So we built this beautiful facility. We load it with Budwood source trees. These trees now, they have to be tested. We have roughly 5,000 trees in this facility. They need to be tested every year for pathogens, not the exotic pathogens that they may have come in from another country. We took care of that once in the lifetime of the variety while the variety was going through the Rubidoux quarantine facility. Now we worry about pathogens that may have snuck into our screen houses by an aphid, local California varieties. Now, as the size of the facility increased and the number of the trees increased, there was no way that we can do those trees one by one and little by little and chopping down every sample with a razor blade. So we moved towards what we call high throughput citrus sample processing. And the traditional way of analyzing, processing a sample to end up with a nucleic acid extract that you can give it to high throughput sequencing or give it to PCR is all this lengthy pipeline you see here with all these specialized pieces of equipment. And it will take us, it was an overnight step. It will take 24 hours. So when we brought up this bottleneck to our university community, Douglas Hill, an engineer at that point graduating with his Ph.D. from the engineering college at UCR, came up with this idea. He said, what if I build you a device that strips out the bark from the Budwood, that is the tissue that you're interested in, and avoid all these steps? I said, go for it. And indeed, Doug, with his partners, built a Budwood tissue extractor protocol, an instrument, but also he built a whole protocol. I'm going to click here and see the video is going to play a little bit, and if I see it, freeze it again.
So the first thing we do.
Yeah, it's freezing a little bit. Let me take the volume down so I can talk to you. He created these specialized bags that they have a little electronic tag on them, and we can scan the sample directly to our mobile device and shoot the name of the variety and the tree location into the cloud in the database. And then without even touching the sample anymore, it gets fed in the machine. And I'm going to move forward over here. In less than 3 minutes, you have this sap in those syringes. And he developed some customized filters that they are in these syringes so we can avoid centrifugations and further pipetting. So this little sample now, this little green sap that it's inside those syringes is ready to be moved forward. And instead of waiting, using roughly, we estimated $120,000 worth of equipment, minus 80s and grinders and waiting for 17 to 24 hours to process roughly 40 samples at a time, within a few minutes, you have this green sap that you can now give to what we call nucleic acid purification methods to extract RNA and extract and purify DNA and RNA and give it to your molecular biology diagnostic technique or pathogen detection technique. But which one are we going to use? There is out there in the market right now, or in the scientific fields, there is 3 different kinds of technologies to purify and isolate DNA and RNA from a green soup like you see here in the syringe. There is a magnetic beads technology, phenolchloroform technology, and silica technology. And the way that you can evaluate how well these technologies perform in terms of isolating nucleic acids is by looking at the concentration of the nucleic acids, the purity, and their integrity. So we started with another PhD student, an experiment where he compared these 3 different technologies, green for the magnetic beads, red for the triazole technology, and blue for the silica and the different manufacturers there, you can see in the graphs. And you could see how concentration for some technologies was higher, but highly variable. You see they have this long standard error, standard deviation bars. Purity was more concentrated, and some cases was better, some cases was not. And integrity, the same thing. So when we put together all this information we had from thousands of samples and so on, the winner for us was this magnetic bead-based technology because it had the highest consistency. It had lower concentrations, but PCR is such a powerful technology, and HTS is such a powerful technology that the low concentration didn't affect the results. But it was very consistent, and it could be automated. So we brought in this MagMarks magnetic bead technology in our protocols at a very critical time around 2009 and 10, when the citrus nurseries were trying to implement the new mandatory program for testing the mother trees in the nurseries, the California Citrus Nursery the California Citrus Nursery Stock Pest Cleanliness Program. So the program before all these innovations could test 500 samples a year, as you can see here with the red bars. So, and the year that the program was implemented, we had to test 7,000 mother trees in the nurseries. So it was impossible. It will take us more than 10 years to test all the trees in the nurseries, the mother trees. So by implementing this high throughput citrus sample technology, you can see the red bars after 2010 explode. We can process essentially unlimited number of samples. So by testing thousands and thousands of samples in the first couple of years, we managed to implement the program successfully. And at the same time, because now we're testing a lot and stay ahead of the disease curve, which a lot of you have heard these principles now with the COVID-19 epidemic, how everybody says, we need to test more, we need to test more. So you need to test fast enough to stay ahead of how a pathogen is transmitted, so you can suppress the transmission and the spread of the pathogen. And that's what the black line demonstrates. When we were testing only 500 samples a year, there was always some viroids, in this case, in our nursery pipeline. After we unleash the high throughput testing pipeline, the viroids essentially disappeared from the nursery pipeline. The other day I had a conference call about what we're going to do next year with CDFA, and they told me the year before they had 0 finds. So this line had dropped down to 0 now. And because this program was so successful, our program received by the American Phytopathological Society a couple of years ago an award for excellence in regulatory affairs. So now you have this system build, you have this mandatory program, system build in terms of quarantine, introduction of varieties, distribution of Budwood into the nurseries that they are operating under this new mandatory program. All production is indoors, minimum risk, and you end up with quality product for the citrus orchards. So this is my last slide where I'm trying to bring all these concepts together for you. So starting citrus correctly and not moving citrus Budwood production correctly and not moving Budwood in our suitcases and bringing it from our friends' backyards or for our international travels and so on is the foundation of the whole system. And in California, we have been trying to do this since the 1930s. So with CCPP and all its partners, the industry and the regulatory partners around them, we produce pathogen-tested Budwood. Then that Budwood finds its way to the nursery industry, that they have done their part with their testing program and their high phytosanitary protocols in their enclosed facilities to produce the best possible tree for the industry. And then the industry, our growers take those trees and put them in the field. But the effort of maintaining, keeping the pathogens and the diseases away doesn't end there. The industry have built these beautiful programs. Examples you can see there, the Citrus Pest Detection Program, and let's see, focus all of us together and see if we can say this acronym, and the CPDPP program. So to make sure that the pathogens are staying away from commercial citrus production. So it's never one test and it's never one idea and it's never one thing. It's always going to be a systems approach. And it always takes all the partners to be involved. So this is how we operate today. And hopefully in the future, when all of us and all the different layers of citrus tree production and citrus fruit production comes under this idea of circular economy, is to take whatever waste we're producing in this pipeline, add value to it and feed it back to the pipeline, not only to have capacity and efficiency, but also achieve sustainability. And as I told you, this is my last slide. I think we're doing well in time. I'm all yours for questions, and many, many thanks for your attention.
Thank you, Georgios. We are going to transition to the Q&A portion. So McCall is going to bring up our first question here. So our first question from George said, "Hi, Georgios, thank you for making the CCPP Budwood available. Do you have a plan to update or include data on the varieties that you offer? The UC Riverside website is incomplete."
Well, it's okay to say terribly. I totally agree with, I think you said George, the person that asked the question. We are trying. We're facing such big challenges all this time with the psyllid coming in and having to prepare the industry to face the spread of the psyllid and the HLB disease, that items such as our website have fallen behind. So right now the Budwood, the variety data link in our website, the ccpp.ucr.edu, on the very top of the site, there is a green link, variety data. When you click there, it takes you, the website reads from our Budwood list ordering system, and it lists all the available varieties from the CCPP. So next to each of the variety, if there is available variety data, there is a link, typically two links, either one that takes you to CCPP data that they were collected from the foundation block when it was active outdoors, or takes you to the citrus variety collection data at UCR. So our plan, we're having these discussions this year with the CCPP committee of the CRB. Our plan is, yes, absolutely, is to invest. Now it's time to start investing on the variety evaluation data and reach our websites and have a more organized system on how to provide this information to the industry. It's not going to be, again, like I hope that message was very clear. All you saw here for the CCPP has been a collaborative effort. It's not going to be CCPP doing it alone. We will do it. We will build the right protocol with the right partners to have variety data available for everybody to see in relationship to Budwood availability.
Thank you, Georgios. The next question is, when will the new HLB resistant rootstocks be available on CCPP? Several have been cleared through the introduction process, but nothing on the CCPP Budwood list. And how will the new pricing relate to those that wish to root the bud sticks?
Yes, thank you for the question. So again, I don't know if you enjoy my presentation or not, but I always like to put things in the historical perspective and why we are where we are today. So very vividly, I remember a 2006 trip with California industry members into Florida discussing these questions. How can we get Florida rootstocks and scions that may be resistant? 2006 was like the year one after the first 2005 identification, but the HLB was spreading in Florida since 1998. So the Floridians could clearly see single trees standing or groves doing better than others. So we in California were anxious, how can we get this material now into California so we can start evaluating and making available and so on. Finally, several years later, the discussions and the paperwork and the protocols and the permits were completed sometime in 2014-15. So finally, we had everything in place for that material to start transferring here. But a key element for us to gain the trust of the system was to say that all this material is going to come into California under this new protocol will be under a material transfer agreement. And that material is controlled and belongs to the Florida breeders and Florida's universities and so on. And we will not do anything in California other than making a Budwood source and waiting for further instructions. So right now, yes, we have created, as we call VI, varieties that they have finished their process and they have a VI number and they have been released from quarantine. They do exist, but right now the industry is in communications with the partners in Florida to develop the protocol, how that material will find its way into orchards and nurseries and backyards and so on. I started from where I started to tell you this is not trivial and easy because it has never been happened before. So, and again, you don't want to make mistakes that create unsolvable issues. So Citrus Research Board, the CCPP, the breeders, everybody's trying to find the right way to do this. One thing that there has been advancement is that some private companies have communicating with the Florida owners and they have set up small-scale field trials. So some varieties have been out there and evaluated in the private sector. We hope that we will achieve the same with the CCPP at the Lindcove station. We have permission to create a mini demo block. We're propagating trees now, and hopefully in the next couple of years, we can take that step. But overall, because we want everybody to understand this and agree on this process, right now we are in the discussions, how do we make this material available into Californians? And we are slow, but it's very important for us to rush it. On the last part, so far the data, since we did a 2 years evaluation of our cost of production of buds. We monitor every single expense that we have, from deferred maintenance of facilities to man-hours to test and cut budwood and package it and so on. We came up with a cost of production per bud at $5.20 for the whole audience to understand the second part of the question. The budwood price of 75 cents was set in 1984. So it had never changed up to a few years ago when we just said for inflation, we're going to make the $1.50. and wait at this price until we have an actual number for cost of production. So when we saw that the cost of production of one bud was $5.20, we updated our price for the non-industry, the non-assessed members. All of you that producing citrus, you're paying an assessment. And that assessment comes back as support to the CCPP. So the people that they were getting the benefit of a subsidized program by the industry, we ask them to pay the real cost of Budwood to bring back some of cost recovery. So far, this happened January of 2022. So far, we have seen a change, not in the number of orders, but how much Budwood is ordered per order. So I don't think the people that want, and that's a general concept as when we're trying to implement the mandatory nursery program, all of us were getting stuck in the conversation about how you can achieve these. People are going to try to do that. You cannot regulate and making programmatic decisions thinking of the people that they're going to cheat. The people are going to cheat, they're going to cheat. So the people that they understand and they want to use the clean Budwood, they have honored and they have completely understood why the price adjustment. The people are going to try to trick the system, they would have tricked it anyway. And they are the people who are going to pay the consequences as diseases are spreading and everybody's doing the right thing. You're not doing, you're sticking up as a sore thumb. And all these beautiful boots on the ground with our survey, the industry survey programs and the CCPP program and the CDFA and the U.S.A, these people are going to serve us and we will know what's going on. So again, the price have changed. We informed the public. We have not seen big changes. I have only a couple months worth of data and we're going to keep monitoring it. And protocols, they're going to be developed for this material to reach its final user and destination in terms of whatever we think may be tolerant or resistant to HLB.
Thank you, Georgios. But we do have a few more questions in the chat. So we actually have a comment from Earl that says we've had nice progress since from 2005 to today. And then Paul Cheney asked, in the case of the Loma Linda Frink adobe trees, is it more correct to remove all of the infected trees and begin a new collection with clean citrus? If new clean trees are planted, will they become infected rapidly from old infected trees?
So for the first part of the, okay, number one, Earl, I'm so happy to see your name there. I'm so happy you heard the presentation. Earl, as he mentioned in his comment, was the first generation, as I call the CRB, as the industry is growing and evolving, has been, going through generations. So Earl was in the first generation. I, as a young scientist, I came into work together. So I'm happy to see you there, Earl. To Paul's question, yes, the adobe, the Frink adobe trees that they are, for the rest of the audience, they are around probably the oldest structure in California or in Southern California. The adobe building was built in 1871. Yes, those trees are heavily infested with graft transmissible pathogens and Phytophthora. They need to be removed completely. After they are removed, the old planting sites, they need to be and removed, including the roots. A backhoe needs to go and pull them out. The planting sites need to be let rest for years if possible and treated against soil-borne fungi and pathogens chemically. And then the new trees that are purchased from commercial nurseries that have been produced through the pipeline we discussed today should be planted not exactly in the same spot, but nearby. And before planting, also need to be treated for, the site needs to be treated for soil-borne pathogens. They may get infected with viruses from aphids and insects if the disease is in the neighborhood, but the risk is no greater than anything else. The most important thing is to remove the whole tree, including roots, sanitize the site properly and prepare the site properly before you plant, and the tree should live very long and healthy lives.
All right, our next question from Tom. How does a selection get selected? What about varieties that never are industry valued?
Okay, from Tom Mulholland. we at the CCPP, because we never see the variety, the fruit, or if it's early or late, we do not know. Our focus at the CCPP and the mission of that first 2005 generation of the CRB that looked forward to the future of the program, we wanted to create a pipeline that can put varieties through without creating a bottleneck, which was huge in the early years, as you saw from my graphs. There was low capacity in the program, it will take too long. So from the phytosanitary regulatory point of view, it doesn't matter if the variety will be planted in 2 backyard trees or in 2,000 acres. We need to make sure we have clean stock for as many varieties as possible, so people won't be tempted to smuggle them into California. The discussion that the question that Tom is asking is a long-standing question, not only for California, but all citrus producing states. So in California, we took the approach I'm telling you about. We want to create a very efficient pipeline so we can clean as many as possible, regardless of their commercial impact. Other programs have committees. They have industry committees that they go through all the varieties that are in the program, and they say this is higher priority than that. And California make such decision as well. So right now, because of the threat of the exotic diseases, and as the HLB3 proved in Los Angeles, there were lemon grafts and pomelo grafts, like varieties that they had no commercial importance for this big scale in California, like navels and mandarins and so on. Nevertheless, these are the varieties that pose the highest risk. So that's the position, and that's how we operate today, and that's how we invest on the program. But if the industry wants to take a different approach and create a priority list, absolutely, this is an industry program and will operate to serve the industry.
Thank you. And we'll just ask one more question here. So George says a very common rootstock in California is C35. How does this rootstock compare to others in HLB resistance? As a home orchardist, I am willing to experiment with new rootstocks on newly grafted trees.
George, I'm not sure I can answer the question. I don't know how much C35 has been used in areas that have been affected by HLB to see how it performs. I suspect because it has trifoliate bloodline, it will cope the infection okay, but this is completely, it's my understanding that trifoliate seedlings themselves, not as only as rootstocks, they're doing better than others when they're infected with HLB, and they have been used extensively as parents to make resistant and tolerant rootstocks. I don't know if I'm looking at my screen, Melinda, from your exposure to all the different research projects, if you have something more for C35 and trifoliate in general for HLB tolerance.
Well, actually, I think what I would do is recommend that the speaker or the audience member attend our meeting on the 28th, which where we're going to have Mike and Tracy Kahn come and talk about all sorts of different varieties. And so they would be an excellent resource to answer that question. Okay.
And really quickly for the last one, Caitlin, which I mentioned already, who is checking if people don't use the right Budwood? Many, many people. There is U.S.A inspectors and CDFA inspectors and all the different programs out there. There is estimated, I think, 50 million citrus trees in backyards, right? It's impossible to know everything. Sick trees will decline and die. So there is a natural evolution there. The problem is that if a vector moves the disease around before the sick tree is dead, you have lost critical time. So I think the outreach and education effort out there is very big, and people do come to the university for Budwood, and nurseries are trying to help and make, specialty trees that they are not, regularly available in retail establishments. it's a constant give and take battle, which its basis and its foundation is education and people making the right decisions. Trying to police everybody, it's an impossibility. And that's why all of us need to spread this message. You want citrus, most likely there is a healthy tree somewhere for you. Do not go and check these resources. Do not put it in your suitcase. That's the message.
Thank you, Georgios. So I'm going to pass it over to Melinda for a few wrap-up comments.
Okay. Yep. No, thank you, Georgios. I would also like to thank Ben Faber for sharing information with us today and our participants for joining us at the Citrus Research Board for today's webinar. CRB is actively planning, along with additional seminars next Tuesday and the following Tuesdays, to see Dr. Jim Ataskevich and Dr. Mike Roos and Dr. Tracy Kahn. We are also planning our annual California Citrus Conference to be held October 19th at the Visalia Convention Center. We hope to see everyone at the conference and will provide further information in the coming months. Thank you so much again for your time.
Thank you, everyone. We appreciate your attendance today and hope you have a great day.